Today we're going to do a review of DNA replication, transcription, and translation. DNA is two-stranded, held together by weak hydrogen bonds. One strand of DNA is made up of nucleotides. There's a nucleotide called A, a nucleotide called T, a nucleotide called G, a nucleotide called C. A nucleotide is made up of the acid, the sugar deoxyribose, which is the backbone, and then the base pair, whether it's the A, C, T, or G. The only part we care about is the base pairs because that is actually the code for the protein that's going to be made. And if you remember, DNA can't directly make the protein. It has to give the code to the messenger RNA, so the messenger RNA and the tRNA can work together to make the protein. But if we need to go through DNA replication, for example, when your cell needs to divide into two smaller cells, the DNA itself is going to have to divide. So every single chromosome in your body is going to have to divide. So an enzyme is going to come along and split the DNA up. So now that it's split, each side can get its new segment. So we would have something like this. I'll do each side individually. There's our two new strands of DNA. In other words, our two new chromosomes. Technically, if you remember, they'd be held together in the center by that centromere. And they would be pulled apart during the phases of mitosis. DNA replication. Then we have transcription. With transcription, RNA polymerase comes along and splits the DNA. Then it will take one strand of the DNA, and we'll use the right-hand side, and it will make mRNA. RNA is one-stranded, not two-stranded, because we're only using one side of the DNA. With the messenger RNA, T becomes A. A's become U's. RNA cannot make the T. C's become G. G's become C. T's become A. C's become G. So now the code that was originally in DNA has been given to the messenger RNA. The messenger RNA will head off to the ribosomes. And in the end, we will have a protein. So then we have translation. This is the newest part of the video. With translation, again, the DNA unsplits. We had messenger RNA made. Now we're going to make an amino acid. Going from DNA to messenger RNA, remember, was transcription. I'm just going to do that really quick because it was up from the previous slide. And again, I'm going to use the right-hand strand of the DNA. And you know you're looking at messenger RNA when you have U's and no T's. You could also know because it's only one-stranded. Now, before we can translate into the amino acids, we have to put them into codons. So messenger RNA has to go into its codons, which is just a group of 3 of the nucleotides. So 1, 2, 3, AUG is one codon, CAG would be the second codon. Now we're going to translate. We're going to make the amino acids that will eventually get hooked together to make the protein. So AUG, you're going to need your translation sheet. Goes to the methionine. And then CAG is glutamine. Now technically, this DNA would keep being read by messenger RNA until it hit one of the stop codons. All the amino acids that were made would hook together and make a protein. So technically we'd have to keep going, but this is just an example. And I think that is it for today. Oh, nope, we have one more practice. Okay, so let's say I gave you some DNA, and I'm just going to write out one strand of the DNA. I'm not going to write out 2 strands, just one strand. Okay? We are going to make some messenger RNA from that. So remember, technically, we would have 2 strands of DNA, but for simplicity purposes, I'm going to give you 1 strand. So we're going to make RNA. We're doing the transcription. We're going from DNA and we're transcribing it. We're rewriting it into the messenger RNA. T's make A's. G's make C's, A's make U's, T's make A, G's make C. Now I can do the translation. Technically I have to put it into codons first. 1, 2, 3, 1, 2, 3. Okay, so remember there's going to be more to the DNA, but we only put it into groups of 3, so we can only make 2 codons in this case. ACC. So now we're translating. We're going from the language of these letters into the language of real words, the amino acids. ACC, threonine, UUA, leucine. And like I said, there'd be really more to this messenger RNA until it hit the stop codon. Threonine, leucine, plus the other amino acids hooked together. We just made the protein. Maybe it was the protein amylase, which helps you break down starch in your body.